Thank you so much for inviting me to give this lecture. I picked this opportunity to present to you scientists our new robotic technology applied to clinical trial fill-finish operations. Our goal is to provide to the research laboratory that are developing new drugs a very simple and reliable tool to improve the quality and the sterility of the product, of the drug that has to be tested afterwards on the patients. Let me say something about our company, Fedegari. Fedegari Group is a company that was established more than 60 years ago, very well known all over the world. We are in the sterilization, steam sterilization business, and in the last 5 years we also have implemented the new technologies, and the technology that I'm presenting right now, it's a clear example of what we are doing. It's a family-owned company, about 400 employees, over 50 million euros turnover. We have seven operations located in Italy, Switzerland, Germany, Singapore, and US. We are designing, manufacturing equipment for pharma, biotech, and food application. One of the key points that distinguish our manufacturing from and design and engineering from other suppliers is that we are manufacture most of our components in-house. So we can say that in our equipment, more than 80% of all the valves and actuators, everything is made in-house. And this is a very important advantage because we have a full control of the technology. These components are designed for our equipment to provide the best technology and the best process. So what we do now? Now we have implemented a new technology. So our vision is to remove all human intervention from aseptic filling operation that will be completely handled by a GMP robotics. It is important for us to share this idea with you guys. You are at the beginning of the new drug development, and according to the quality by design approach that was focalized by FDA a few years ago, we need to build up the quality since the early beginning of the design of the drug, and also the process that is required to manufacture the drug. So it's a complete, full combination of two different approaches, but having the same denominator that is quality. We have to build up the quality. And this system that we have designed is a typical example of how to build up the quality in a fill process. So the outlook of the pharma business, we have a lot of patents that are expiring or were expiring the last years. So there is quite a huge amount of investment in research and development. There is a lot of pressure also in anti-cancer drug. So the demand of anti-cancer drug is growing rapidly. And out of 100 ongoing clinical studies, more than 65 are for anti-cancer drug. And also biopharma research is a relevant source of new active principles. And what is important for our technology that more than half of the newly developed drugs are injectable. So they have to be manufactured in sterile conditions. Who is it for? We focused high-value niche therapies. I mentioned just some of them. Monoclonal antibodies, gene therapeutics, orphan drugs, recombinant proteins, drug combinations. What do they have in common? They have in common that they are personalized drugs, so small batches. Therefore, we will have shortened run to manufacture this drug, but they have a greater complexity and also a more volatile demand. As I told you before, there is a lot of quality expectation in this drug. Consider that developing this drug, for instance, to get just few milligrams of an active principle, maybe it takes some months, years, you know. If you don't handle this principle in a very safe condition, there is a high risk to lose it because of contamination of your batch. And that means that you throw out of the window a lot of work to produce again the active principle ingredients. So it is really a very challenging process. The core of the issue, as most of you know, is the human being. Human being is generating millions of particles, viable and non-viable, and all these particles are potentially a risk for your sterile manufacturing. So if we find a solution in which in this equation the human being is out of the equation of manufacturing, then we will for sure achieve what my friend Jim Magalloco says an advanced aseptic process. An advanced aseptic process is a process in which there is no need of human intervention. So why a robotic aseptic filling solution? Flexibility, modularity, and scale-up manufacturing. It means that if we start to develop with this system a new drug, in a few months we can scale up the process for a larger manufacturing. It is designed for multi-format. So it means that with this system you can handle vials, syringes, pre-filled syringes, and cartridges. So that are the most used formats used for supplying the drugs to the patients. And furthermore, we are talking, these drugs are they have a high level of toxicity. We are talking of a toxicity level that is a grade 4 and 5, according to the occupational exposure band. We are talking of a toxicity of impact of nanograms of these drugs against the human health. So we need to have a system that completely isolated these high toxic drugs in order to run a very safe process, also environmental safe process. Compact equipment, so the footprint of a unit like this is more or less 4 meters by 2 meters by 3 meters tall. So it can stand in all laboratories. And the need is just for an ISO 8 clean room that is a very simple clean room, not sophisticated as the clean room for aseptic manufacturing. And what is important also is that FDA and EMA recognize, I had some talks with some FDA inspectors presenting our concept, and they were excited because they are pushing all the pharmaceutical companies to isolate the process both with restricted access barrier system or isolation technology. So in this animation you can see what we are talking about. We have the robotic arm. we call a snake robot. It is an anthropomorphic arm with 7 degrees of freedom. So on the right side, we have an autoclave that makes the sterilization of the primary containers, like the vials or the syringes or the cartridges. And these are held in this tray. This tray is closed with a Tyvek sheet. So the robot is able to remove, and we'll see in a real simulation, to remove the Tyvek lid, and then to handle all the trays in the different position. We have 3 pedestals, one for stoppers, one for plastic seal, and the third one for the vials. What is important also is that this robot, according to the GMP requirements, you need to make environmental monitoring, so to check the sterility of your environment. So to check the sterility of your environment, you have to use active and passive sampling, biological sampling. So the robot can handle also this Petri dish for active sampling and passive sampling, and then also for swabbing. So you have to swab the critical surface to check that there is no contamination on the critical surface. So we are almost arrived to loading the containers, the vials, and then the design, we have a gripper that can grab different tools. So according to the phase of the process, this gripper moves to grab different tools. In this case, the gripper has some fork to move the tray into position. And then afterward, you will see that the gripper will grab the filling needle and start the filling operation. And the same can grab a vacuum pad for a pick and place of the stoppers and the seal. So you see now he's making the filling operation. So small batch, what does it mean? It means a few hundreds up to 1,000 vials, 10 milliliter vials. That is more than enough for preclinical phase and clinical trials of up to phase 3. And here the vacuum pad grab the seal and make the seal, clip in the plastic seal and perfectly seal the vial. Okay, with this process, we can say that we can improve the sterility assurance level of the sterile manufacturing. The isolator, as I told you before, is fully sealed. We can control also, we need some other tools and system to be introduced into the isolator. And we can use what is called rapid transfer port. So there are kind of door that connected to half door connected together. They keep the contamination inside the 2 doors, and then can be open into the sterile environment. And I can transfer in a sterile way the material inside the isolator. Also, to introduce the drug that has to be filled, there are also fast connection, sterile connection, quick connection that you can make even in an environment like this with the full guarantee of the sterility. What is also important is to mention that we don't have any gloves to work inside the isolator. Gloves are a source of issues because dealing with glass, you can get punctures, And then after you discover this puncture and these leaks just after you have completed your batch. And if you have a puncture, then you have to consider the possibility to throw away all the batch, to refuse the batch. It is also important, as I told you before, the safety of the operator. We are handling very high active ingredients. The isolator is completely sealed. We make a pressure test to check the airtightness of the system. And the unit is classified according to the US GMP ISO 5 and Class A according to the EU GMP. And the isolator can provide also a washing place, so all the spray generated during the filling operation of your product can be washed down and inactivated by this washing place system, so you can safely open the door when you have completed the operation. We have also to take in consideration the highly cost of product loss. So we are handling tray by tray. Generally, a tray contains from 100 to 60 vials. So if there is any issue, we are losing just a part of the batch, not all the batch. So, you know, the system has been evaluated by the risk assessment, and we have emergency gloves for make emergency operation. What is important? What is you know, the main problem to lose a batch? Lack of sterility generated by poor aseptic techniques. Your operator has to be fully trained and retrained every few months. They have to be retrained and rechecked if they are properly working. You know, with a robot, you train only once. And then for the life of the robot, he will do the same every time. Cross-contamination because of lack of integrity of the equipment. So if you have some leaks, then you can get some cross-contamination. Operator mistakes, equipment breakdown. So I would say that this solution minimizes the risk of losing product. We have also introduced for this solution single and ready-to-use material, so we don't need to spend time and you know, and time for cleaning, a lot of time for cleaning, and also a lot of time for cleaning validation, especially with these high potent products. Cleaning validation could be a nightmare because you need to remove all the traces of your drug up to level very, very low. So it's also difficult to detect. And then using a single-use system, the change from one batch to the other batch is minimized. So we have a very high uptime for this equipment. We already talked about glass breakage. So with this moving the glassware through trays, we are moving the trays, not we are moving each single vials. And the risk of a glass breakage is almost minimized. More confidence in facing regulatory risk. You know that you have to be evaluated by inspectors when you are making clinical trials as well. So if a regulatory body understands that you have your process fully under control because of this robotic solution, then the scrutiny would be for sure less than with making a manual operation. A lot of scrutiny with the manual operation. Robotics, just a few words about robotics. Normally, you know, first of all, robotics were introduced since, I would say, 40, 50 years ago in automotive industry. So the robotics has developed a very reliable it's a very reliable equipment, so very well known. The difference is the material. So this is a material that is not very good for chemical biodecontamination, because we are decontaminated this all the isolator and the robots with vapors of hydrogen peroxide. So we burn with hydrogen peroxide all the viable matter that we have inside the isolators. And in the last 3 years, we spent 3 years' time to develop together a Japanese company this solution. It's a fully stainless steel robotics, completely airtight. So these are the rotary gaskets that make the tightness of the system. It has 7 axes, so 1 axis, 2 axes, 3 axes, 4 axes, 5 axes, and so on. This robot can make movements that with your arm you can't. Otherwise, you broke your shoulder. And here you can see an example of this robot. Okay. Since we need the human being to make the programming of the system, so we need a higher level. So, just to give you a quick idea, we can have an output rate of between 12 to 20 vials per minute, based on 10 milliliters format. So in our equipment has been implemented steam sterilization for the primary container, hydrogen peroxide decontamination for the chamber of the isolator. So we can make clean in place, wash down in place. There is no change parts. Changing from one format to the other formats is just a question of teaching the controller with the new softwares, size number 1, size number 2, size number 3. No glove posts, and a lot of use of single-use material. These are the nested and trade vials, the format. They come from the manufacturer already sterile, so you don't need you need just to make an outer sterilization of the container. And this here are just a few other simulations. So in this case, we didn't have yet the stainless steel robot, but just to show you the autoclave. And here, when we got the new robot, we make some tests to remove the Tyvek lid from the tub, from the tray. You see the carousel holding all the containers. So now we are in a further stage. So I guess that within this year, the first unit is ready to be on the market. So I took this opportunity also to share with you our experience and understanding if a solution like this can help you in your work. As a summary, we can say that nothing is advanced with human intervention. Don't be scared to redesign the aseptic process. We can also say that complexity is the enemy of reliability, so we need to have a very simple solution, but extremely reliable. We don't want the gloves. Less moving parts means less problem and particles, and we need to look toward to have new approaches to the process. Thank you very much for your attention, and ready for questions. Almost on time, 25 minutes.
If you have we are running with a shortage of time, but still if you have one question. Yes, please.
Yeah, you pick the point, because our idea is also to provide the possibility to rent such kind of machine. But in any case, just for your information, our strategy, and we are on this track, having a pharmaceutical partner to make all the, I would say, the pharmaceutical development, pharmaceutical process development, together with us in order to comply to all the FDA and the EMA requirements.
Yes. Sir, one simple question. Will this process ultimately lead to increase or decrease the overall cost of the product, final product?
Sorry, say it again because I couldn't.
Please give the microphone.
Will this process ultimately lead to the increase or decrease of the final product? Cost factor.
Cost factor.
The cost factor. Okay. The cost factor. You make an excellent question. The cost factor is really not. You have to consider what is the cost of your batch. There are some drugs that a batch cost millions of dollars. So of course this unit has quite a high relative impact. But if you lose a batch that costs you $1 million because you are using poor aseptic technique, what is the cost for you not having this system? So we are talking of a cost between 700 to 2 million euros.
No, I just wanted the cost for the customer, ultimately.
The cost for the customer, the patient, you are saying?
Yeah, means will it, will he has to pay more or will he has to pay less?
If we were using that robot,
No, it will not increase in the cost. It will not increase in the cost because your developing time will be reduced because you will skip all the risk of losing productivity, and you can get much faster to the patient that are on trials. So at the end of the story, if you can speed up the development process, then this absorb the cost of this system.
Thank you very much for this interesting talk. Maybe there was a confusion about the fact that this is for manufacturing purposes or for the clinical trials. But even for the clinical trials, part of the process that you presented was handling the sterilization, the autoclaving, and all that stuff. Does this machine also process the water? Because the water for injection and all that stuff as well is part of the game.
Okay. I would say that there are water for injection, there are different systems to manufacture water for injection. You can fill and then make a terminal sterilization. So you are really sure that with water, it's 100% sure that no contamination. into the water.
So does the system produce water for injection, or you have to buy that?
No, this system doesn't need water. Need the water for injection just for cleaning purpose. You know, this system manufactures in sterile condition some drugs that cannot be terminally sterilized. You know, if you cannot, and most of the biological and large molecules cannot be terminally sterilized. Water for injection can be terminally sterilized. So if you go to the, have a quick review of the GMP good manufacturing practice, all the organization, FDA and EMA, they state, as soon as you have the possibility to make terminal sterilization, please do, because terminal sterilization is give the highest safety for not to having biological contamination. But because here we are handling drugs that cannot be terminally sterilized, and we are talking of drugs that have a very high cost. You know, water for injection is a very poor material, you know. Okay? Thank you so much again for a lot of questions.
In fact, in this 21st century, this the century of technology and this computer engineering and robotic engineering has merged with this pharmaceutical technology and a lot of improvements. In fact, this cost factor, if this technology and this machinery are available suitably, as we face also earlier this capsule or tableting technology, so many instruments are used. What is the cost factor? So similarly, if these things are available, it will be cheaper. Anyhow, it's an excellent job, and in fact, we are in the robotics age. So, excellent work done by this Professor Sergio Mauri from Italy. Thank you for his appreciation of this work. A certificate for him.
Okay, thank you so much. I'm very glad to be here.